Parkinson's and Alzheimer's diseases, which affect a large portion of the global population, are examples of accelerated aging. These conditions cause movement and cognitive difficulties, often with overlapping symptoms. Research into aging has focused on amyloids, solid-like protein aggregates. Neurologists refer to the amyloid state as pathology. Notably, most people form amyloids as they age, yet only a small fraction develop neurodegenerative diseases. Amyloids are formed when proteins misfold and clump together. This so-called misfolding of proteins is part of a change in the structure of proteins, from their native soluble and functional state to their insoluble amyloid state. The core structure of amyloids is a cross-beta pleated sheet, which is among the most stable configurations in nature, making it unlikely to inflict toxicity. When amyloids form, proteins are no longer in their normal, soluble, and functional state. In healthy aging, the brain is able to replenish the levels of normal proteins. In accelerated aging, the replacement does not match the consumption, and the level of the normal proteins drop. The larger the deficit of normal proteins, the more severe and more rapidly progressive the disease is. Proteins shift from the soluble and functional to the insoluble and non-functional state through a process called phase transition, like how water turns to ice. This requires specific pH, temperature, catalysts, and high concentrations, supersaturation. In the physiological state, there is an energy barrier called the nucleation barrier that slows this process down. If we add more proteins or raise the concentration, this barrier gets lower, making it easier for proteins to change from their normal state to an abnormal one. A better understanding of the physics of protein transition to amyloids, as presented in this BioEssays review, permits formulating new strategies for combating accelerated aging.